Part-2 Molecular Basis of Inheritance. 5.4 Replication. While proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: "It has not escaped out notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." Watson and Crick, 1953. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each of each of a molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-consecutive DNA replication, figure 5.6. The experimental proof: it is now proven that DNA replicates semi-consequently. It was shown first in Escherichia coli and subsequently in higher organisms such as plants and human cells. Matthew Masselson and Franklin Stahl performed upon the following experiment in 1958. They grew Escherichia coli medium containing NH4Cl. The N15 is a heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that the N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation. Now, in cesium chloride is CSCl density gradient. Please note that N15 is not a radioactive isotope and it can be separated from N15 is not a radioactive isotope and it can be separated from N14 only based on densities. The second, then they transferred the cells into a medium with normal NH4Cl and took samples at various definite time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. Various samples were separated independently as CSCl gradients. Now, to measure the densities, cesium chloride gradients are to measure the densities of DNA, figure 5.7. Can you recall what central goal the force is and think why a molecule with higher mass density would sediment faster? The result shown in figure 5.7. Third, thus the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes. The Escherichia coli divides in 20 minutes, had a hybrid. Had a hybrid or intermediate density DNA extracted from the culture after another generation, that is after 40 minutes into the second the generation was composed of equal amounts of this hybrid the DNA and of light DNA. Thus, if Escherichia coli was allowed to grow for 80 minutes, then what would be its proportions of light and hybrid densities? DNA molecule very similar. Experiments involving use of radioactive thymidine detect her distribution of newly synthesized DNA in the chromosomes was performed on Vicia beans by Taylor and colleagues in 1958. The experiments proved that the DNA in chromosomes also replicates semi-conservatively the machinery and the enzymes. The living cells, such as Escherichia coli, the process of replication requires a set of catalyst enzymes. The main enzyme is referred to as DNA dependent DNA polymerase, since it uses DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have catalyzed polymerization of a large number of nucleosides a very short time. Sclerocola that has only 4.6 into 10 is to 6 BP. Compare it with human food. Deployed content is 6.6 into 10 is to 9 BP. Completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 BP per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction and this with higher degree of accuracy. Any mistakes during her application would result into mutations. Furthermore, energetically, replication is a very expensive addition to acting as substrates. They provide energy for polymerization reaction. The two terminals of phosphates in the deoxyribonucleoside triphosphates are high energy phosphates, same as in case of ATP. In addition to DNA dependent DNA polymerases, many additional enzymes are required to complete the process of replication with with a high degree of accuracy for long DNA molecules. Since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occurs within a small opening of the DNA helix, referred to as replication fork. The DNA dependent DNA polymerases catalyze polymerization only in one direction, that is 5 prime to 3 prime. Creates some additional complications at the replicating fork. Consequently, on one strand, the template with polarity 3 prime to 5 prime, the polarity 5 prime to 3 prime, it is discontinuous. The discontinuously synthesized fragments are later joined up by the enzyme DNA ligase. Figure 5.8. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite region in Escherichia coli DNA where the replication originates. Such regions are termed as origin of replication. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vectors provide an origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and cell division cycle should be highly combined. A failure in cell division after DNA replication results into polyploidy polyploidy or DNA replication results in a point to a polyploidy chromosomal anomaly. Now you will learn the details, nature of origin and the processes occurring at this site in higher classes. 5.5 transcription, the process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Here also the principle of complementary governs the process of the transcription, except the adenosine the complementarily governs the process of transcription, except the adenosine complements now forms base pair with uracil. instead of thymine. However, unlike in the process of replication, which once set in the total DNA of an organism gets duplicated, the transcription only a segment of DNA and only one of the strands is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed with why both the strands are not copied during transcription has the answer hence the simple answer first in if both strands act as a template they would code for RNA molecules with the different with different sequences. Remember, complementarily does not mean identical, and in turn, if they code for proteins at the sequence of amino acids and the proteins would be different, hence the segment of complicate and the genetic information transformation is second. The two RNA molecules of if produced simultaneously would be complementarily to each other, hence would form a double stranded DNA. This would prevent the RNA from being translated into protein, and the exercise of transcription would become of for futile one. Transcription unit then unit transcription unit is in the DNA is defined primarily by the three regions in the DNA. First a promoter, second the structural gene, third a terminator. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit, since the two strands have opposite polarity and the DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5 prime to 3 prime. The strand that has the polarity 3 prime to 5 prime acts as a template and is also referred to as template strand. The other strand, which has the polarity 5 prime to 3 prime and the sequence same as RNA except thymine at the place of uracil, is displaced during transcription strangely. Now this strand which does not code for anything is referred to as coding strand. All the reference point while defining a transcription unit is made with coding strand to explain the point on the hypothetical sequence from transcription unit is represented below. The promotes and terminator flank are the structural gene in a transcription unit. The promoter is said to be located towards 5 prime end upstream of the structural gene. The reference is made with respect to the polarity of coding strand. It is the DNA sequence that provides a binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding stands by switching its position. Now with terminator, the definition of coding and template stands could be reversed. The terminator is located towards 3 prime end downwards of the coding stand and it usually defines on the end of the processes of transcription. Figure 5.9, there are additional regulatory sequences that may be present further upstream or downstream to the sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with the regulation of gene expression. Transcription unit and the gene is defined as the functional unit of inheritance. Through there is no ambiguity yeah, that the genes are located on the DNA. It is difficult to literally define a gene is in terms of DNA sequence. The DNA sequence coding for tDNA and rDNA molecule also define a gene. However, by defining a cistron As a segment of DNA coding for a polypeptide. The structural gene is transcription unit could be said as monocystonic, mostly in the eukaryotes. The transcription unit could be said as monocystonic, mostly in eukaryotes, or polycystonic, mostly in bacteria or prokaryotes. In eukaryotes, the monocystonic structural genes have interrupted coding sequences. Now, the genes in eukaryotes are split. The coding sequences are expressed. Sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exams are interrupted by introns. Introns or intervening consequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of DNA segment. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though the sequences do not code for any RNA or protein types of RNA. And the process of transcription in bacteria, there are three major types of RNAs: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All three RNAs are needed to synthesize a protein in cell. mRNA provides the template. tRNA brings amino acids and reads the genetic code. The rRNA plays the structural and catalytical role during translation. There is a single DNA-dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription. Initiation need is just nucleoside triphosphates as substrate and polymerizes in a template dependent fashion following the rule of complementary. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, there's a nascent RNA falls off. Now, so also the RNA polymerase. This results in termination and transcription. And intriguing question now is that how is the RNA polymerase able to catalyze all the three steps, which are initiation, elongation, and termination. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor and on termination factor to initiate and terminate the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. Figure 5.10. The bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation take place in the same compartment, there is no separation of cytosol and nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. So consequently, the transcription and translation can be coupled in bacteria. In eukaryotes, there are two additional complexities. There are at least three RNA polymerases in the nucleus, in addition to the RNA polymerase found in the original organelles. There is a clear cut division of labor. The RNA polymerase 1 transcribes rRNA to 20S, 18S, and 5.8S, whereas the RNA polymerase 3 is responsible for transcription of tRNA, 5S RNA, and snRNAs, small nuclear RNAs, and RNA polymerase. Second, transcribes precursor of mRNA, the heterogeneous nuclear RNA, hnRNA. The second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. Hence, it is subjected to the process called splicing, where the introns are removed and exons are joined in a defined order. HNRNA undergoes additional processing called as capping and telling. Capping and additional processing called the capping and tailing. In capping, an nucleotide methyl guanosine triphosphate is added to the end 5' end of hnRNA, and in tailing, the adenylate residues 200 to 300 are added at the 3' end in a template independent manner. It is a fully processed hnRNA, now called mRNA, that is transported out of the nucleus or transcript or nucleus for translation. Figure 5.11 in the significance of such complexities now is how now beginning to be understood. The split gene arrangements represent probably an incident feature of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In recent times, the understanding of RNA and RNA dependent processes in the living system have assumed more importance. 5.6 genetic code. During replication and transcription, the nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. Now, the process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize, transfer of genetic information from the polymer of nucleotides to synthesis a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There existed ample evidences to support the notion that change in nucleic acids genetic material were responsible for change in amino acids, other proteins. This led to the proposition of genetic code that could be a direct the sequence of amino acids during synthesis of proteins. In determining the biochemical nature of genetic material and the structure of DNA was very existing exciting. The proposition and deciphering of genetic code were for most challenging. In a very true sense, it required the involvement of scientists from several disciplines, physicists, organic chemicist, biochemist, and geneticist. It was George Gamow, a physicist, who argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because of permutation of combination of 4s to 3 4 power 3 4 into 4 into 4 would generate 64 codons. Generating many more codons than required provides proof that the codon was a triplet was a more daunting task. The chemical method developed by her Govind Khurana was instrumental in synthesizing RNA molecules with defined combination of bases, homopolymers and copolymers. Marshall Nirenberg, the cell-free system for protein synthesis, finally helped the code to be deciphered. Serostia enzyme polynucleotide phosphorylases was also helpful in polymerizing RNA with defined sequences in a template independent manner. Enzymatic synthesis of RNA. Finally, a checker board for genetic code was prepared, which is given in table 5.1. The codons for the various amino acids. The certain features of genetic code are as follows. First codon is a triplet. 61 codons code for amino acids, and 3 codons do not code for any amino acids. Now, hence they functions as stop codons. Second, some amino acids are coded by more than one codon. Hence, the code is degenerate. The codon is read in the mRNA in a contiguous fashion. There are no punctuations. Fourth, the code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine, PHE. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. Fifth, AUG has dual functions. It codons for methionine, MET, and it also acts as initiate or codon. 6 UAA, UAG, UGA are store terminator codons. Now, it following is the sequence of nucleotides in mRNA. Predict the sequence of amino acids by it. Take help of the checker board. AUG, UUU, UUC, UUC, UUU, UUU, UUC. Now try the opposite. Following the sequence of amino acids coded by an mRNA. Predict the nucleotide sequence in the RNA. MET, PHE, 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 PHE. Do you face any difficulty in predicting the opposite? Can you now correlate which two properties of genetic code you have learned? 5.6 Mutations and genetic code. 5.6.2 tRNA, the adapter molecule. 5.7 Translation. 5.8 Regulation of gene expression. 5.8.1 the lac operon. 5.9 Human Genome Project. 5.9.1 Saline features of human genome. 5.9.2 Applications and future challenges. 5.10 DNA fingerprinting. देन समरी. मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस में हमने पहले से देखा स्टार्टिंग से. द DNA, structure of polynucleotide chain, packaging of DNA helix, the search for genetic material, biochemical characterization of transforming principles. The genetic material is DNA, RNA world, replication, the machinery and the enzymes, transcription, genetic code, mutations and genetic code, translation, regulation of gene expression, the lac operon, Human Genome Project, saline features of human genome, applications of future challenges, DNA fingerprinting, then summary. Please like, share and subscribe. Thank you so much for watching.